Hey everybody, I'm Dr. Wyrick. I'm the Clinical Director of Intermountain Regenerative Medicine. And what I'm going to share with you today, I promise you, is going to be the most boring video that I will ever make. But because what I'm going to do is I'm going to get into a little bit of the science and some of the genes that are expressed on stem cells that need to be there in order for us to call a stem cell a stem cell. So we're going to get into some really minute details here for the next few minutes. So bear with me. But let me just go through what these markers are, what they mean, and why we're even talking about them. So that being said, we're going to jump into this. November 2020 is a very important day in our industry. And the reason why it's an important day is because that is the day, it's November 30th, 2020, excuse me. That is the day that the FDA has chosen as the cutoff line for stem cell therapy. And what that means is this. On November 30th, 2020, labs, clinics, and practitioners that are doing unscrupulous things, they've got until that day to get their act together before the FDA starts coming after them and cracking down on them. So this is actually a really, really good thing for our industry, because what it's going to do is it's going to separate the wheat from the chaff, and I'm actually very excited about it. The information I share with you will keep us on that side of the fence, so we're super excited about that. But it is going to be a good day. So this, what I'm sharing with you today, is part of the FDA's process for us to be able to stay on the wheat side versus the chaff side. So we're going to get into that. But this International Society of Stem Cell Therapy, International Society of Cell and Gene Therapy, has a set of standards that they've come up with that need to be followed in order for us to call a stem cell, our product, a stem cell. So I'm going to go through with you what that list is. This is where it gets really boring. So these are the attributes that mesenchymal stem cells have to show in order for us to be able to call them a mesenchymal stem cell. So number one, they got to be from a biologically young source. So that source, of course, is going to be the umbilical cord. I've got in other videos, I've gone into the details of that. They have to positively express the following genes. CD stands for cluster differentiation, but they have to positively express CD73, CD90, and CD105. We're going to talk about what that means in this video. They have to have a lack of expression of these following genes in order for us to call them a stem cell, and those are CD105, CD14, CD34, CD45, and HLA-DR. So we're going to talk about what those mean, so let's get into it. So a positive expression of CD73, that means that they have to express this cluster differentiation 73 in order for us to call a stem cell. What CD73 does is it converts AMP into something called adenosine. Adenosine is the building block to ATP, which is how your body makes energy. So if you think about it, if we're going to grow new tissue, create new cartilage, those cells have to have the ability to produce their energy if they're going to survive and be viable. So in order for us to call a mesenchymal stem cell, CD73 definitely has to be expressed. The next one is going to be CD90. Again, these are the ones that have to be expressed. CD cluster differentiation 90 is a positive marker that the cell is indeed a stem cell, meaning that stem cell is a blank slate cell that can become other tissues that the body needs it to become. And then lastly, they have to express this CD105. CD105 plays a crucial role in what's called angiogenesis, which is the formation of new blood vessels. So if you think about it, not only does a cell have to be able to produce energy, but you have to get fuel to that cell to produce energy. And if CD105 is present, then then it'll be able to grow a new blood supply to that cell, and that will maintain a viable cell. So lack of expression. These are the cells, these are the genes that they cannot express in order for us to call it a stem cell. Hey, CD105 is a hematopoietic cell, meaning it's going to form blood cells, not blood vessels. So we can't express those. CD14 has to do with your innate immune system cells. So if it's expressing CD14, there's a higher probability that this is going to create an adverse event, something like an autoimmune type of reaction. CD34 is another hematopoietic cell that cannot be expressed in order for it to be called a stem cell. And then there's CD45. CD45 is an immune function. It's a regulatory B cell and T and B cell antigen receptor signaler. Meaning if CD45 is going to be expressed, it's going to mess up your T and B cell regulation, which is an immune system function and can lead to immune system problems down the road. And then last but not least is this gene HLA-DR, which is a gene that's been linked to a lot of host versus graft, in other words, rejection, host versus graft diseases, and other autoimmune issues. So we definitely don't want those expressed. So if this product that we're using, if the stem cells that we're using hit all of those requirements, then you're actually getting a mesenchymal stem cell. This is why we're very particular about the labs that we use. And if you choose not to do stem cell therapies with our clinic, please make sure that the one you use is also following those same standards. So thank you. I know that was the 5 minutes of the most boring video that I've ever made. We hope you enjoyed, and we hope to talk to you very soon. Thanks a lot.